Good afternoon or good morning, everybody, depending on where you're joining us from. Thank you for taking the time to log on today. We have a great talk lined up with Dr. Ken Margulies, who is joining us from the Perelman School of Medicine at the University of Pennsylvania, where he is a professor of medicine. And he is going to be talking to us a bit about some basic physiology regarding or involving the cardiac myocyte, which is a testable topic for those of you that are looking forward to taking your heart failure boards in the future. So this is very important because as I recall, there were some questions in this domain on the board exam. So Dr. Margulies, thank you so much for taking the time to put this together for us, and we look forward to hearing what you have to say.
Terrific. Happy to join, and let's get started. So again, on my topic here is excitation-contraction coupling. Let's begin with reviewing the learning objectives. By the end of this, you should understand how an electrical signal, action potential, within a cardiomyocyte triggers changes in calcium movements, leading to the physical contraction of the muscle fiber. So that's the essence of excitation-contraction coupling. As part of that, you should be able to identify structural and molecular constituents of cardiomyocytes that are most relevant to excitation-contraction coupling, including the sarcolemma, transverse tubules, or T-tubules, sarcoplasmic reticulum, and the contractile elements and associated proteins. And particularly understand the roles of various associated proteins as listed in the next block of text. And at the end, we'll take a moment and pull back a little bit and appreciate how contractility is graded at the cardiomyocyte level by cell length, stimulation frequency, and beta-1 adrenergic stimulation. So let's get started. Let's begin with a cardiomyocyte. This is a very cardiomyocyte-focused talk, and so this is a picture of an isolated rat cardiac myocyte following collagenase digestion. You can see there's a couple of nuclei sort of shown here, lots of stripes. The striated pattern is caused by longitudinal arrangement of sarcomeres in a very organized fashion that we'll delve into a little bit more. One of the things I want to highlight, even at this first slide, is that myocytes are big cells compared to other cells around the body, much bigger than white cells, and considerably bigger also than hepatocytes or epithelial cells or nearly any other cells around the body. Skeletal muscles are an exception that are bigger still, but they have some of the same adaptations to manage their excitation-contraction coupling needs, as we'll go into in a moment. If we drill down a little deeper, this is a cartoon based on electron microscopy level magnification of a portion of an isolated myocyte. And what's highlighted here is that it's packed with myofibrils, shown here in a very organized sarcomeric pattern, that between every set of myofibrils is lots of mitochondria, because these are very energy-consuming processes. That won't be our focus here, but it's worth recognizing that the contractility work is very closely linked to the energetics needs. And one thing mentioned here in this picture is a T-tubule. This is an invagination of the plasma membrane. This is a surface membrane, and that it goes deeper into the cell, even deeper than shown here. And we'll get back to that in a moment. But this is a consequence of these being very large cells. And if you're going to activate myofibrils in the interior of these relatively thick cells and large cells, you have to have lots of ways of getting the plasma membrane electrical signal into the interior of a large cell. I also want to point out that stripe pattern comes from a very organized sarcomeric pattern. In fact, it's related to the overlap or non-overlap of myosin, the thick filament, and actin-containing thin filament. We'll go back into those a little further in this talk. I want to also highlight the magnitude about, how many sarcomeres and how many cells we're talking about here, right? So in a given myofibril, which is just one of those stripes within the cells on the previous slide, there's hundreds to tens of thousands of sarcomeres. And within every there's also hundreds to tens of thousands of myofibrils for each cardiomyocyte. And then there's about 7 billion cardiomyocytes per adult human heart. So when we talk about sarcomeres, we really are talking a very small structural unit of a very macro level structure. This is a slide I'm going to be using again and again. It's related to one of the readings that I've associated with talk. Don Beers is one of the sort of kings of calcium and an expert in excitation-contraction coupling, literally has written the textbook that most people use on this. I don't think for the boards you're going to need to read the textbook or even the full article, but the full article is interesting and comprehensive, even though it's almost 20 years old. So what I'm going to highlight here is the major portions of that contribute to the dynamics of excitation-contraction coupling. In this cartoon, we see the plasma membrane on the surface in white here, the sarcolemma. And then the invaginations of the sarcolemma that I pointed out on the previous slide, that's a T-tubule. And again, the main functional role there is to bring electrical impulses from surface ion channels down into the interior of the cell. More on that in a moment. There's a number of those ion channels shown on either the surface plasmalemma or the T-tubule sarcolemma. And one of the things that we'll highlight is that some of these proteins, like the L-type calcium channel highlighted here, are in really close proximity to internal proteins like the ryanodine receptor. And so that their spatial proximity is purposely drawn very, very close together here. And this is called the sarcoplasmic reticulum, internal calcium storing organelle. Has other functions, but that's the biggest one. And so there's calcium coming in from the outside, and there's also calcium being released from the inside. We'll get more into that later. One of the other things you see on this cartoon is myofilaments. And as you can see, this is an oversimplification. We saw in the previous pictures that the cell is really packed with myofilaments. It's not just one little myofilament in a sea of cytosol, as shown here. The other thing I want to highlight here, and I'll mention again, is that there's a cascade of events, right? The first is the electrical event, the action potential. We'll go into that in a little more detail. Then there's a released calcium event. We call that the calcium transient increase in intracellular calcium and then a decay. And then finally, the contraction is a consequence of those increases and decreases in calcium. So the increases in calcium yield increases in contractile force. And as the calcium is decaying, waning in its concentration inside the cell, then that in turn, with some delay, triggers a reduction in contractility of force of the cell. So beginning with excitation part of excitation-contraction coupling, I think this will be familiar to most here, that there's action potentials throughout this excitable tissue we call the heart. Some of them are pacemaker cells, like in the SA node shown here, and those have a particular wave shape, particularly this phase 4 depolarization, or here it is in the SA node, phase 4 depolarization, that leads to intrinsic pacemaker activity. It's not going to be our major focus here, but we also recognize that further down in the cardiac conduction system, and in fact within all the cells of the heart, there are more characteristic action potential wave shapes as shown here. So the ventriculomyocytes comprise the biggest cell population of the heart, and their typical action potential wave shape looks something like this. Not so much intrinsic pacemaker activity, it's pretty flat in phase 4, but a rapid upstroke, a plateau phase, and a decay before we begin it all again. And again, one of the things to remind this clinically oriented audience is that all of this is reflected on the surface ECG, which in some ways is a histogram of the billions of cells and their depolarization. So the cells of the atria and their depolarization and repolarization are reflected in the P wave, the depolarization of the ventricles and the QRS complex. And again, all these repolarization events are reflected in the QT interval. I want to mention that these action potential wave shapes are the summative result of many, many different ion channels shown here. I'm not going to go over them in detail, and you won't be tested on them, but it's suffice to say there's a few that you should know a little bit about. One is that the main thing leading to this rapid upstroke of either the atrial or the ventricular action potential is the sodium current. And it's a massive increase in conductance that leads to that change from a polarized resting potential to a depolarized active potential that opens up a lot of ion channels, as you see here. And again, we won't go into the details, but suffice to say, it begins a calcium entry, and that the L-type calcium channel is the most prominent calcium channel affected here. And also a lot of potassium channels are shown here. Again, that pacemaker current, when it's applicable, is often shown as IKF, and that's shown here. Now, one of the ways to sort of summarize all those individual ion channels is shown in this old slide, but it gets the point across that in a ventricular myocyte, this is a very characteristic wave shape. What we see is a big influx of sodium that's responsible for the rapid upstroke. For the most part, there's a dynamic equilibrium going on during the plateau phase of the action potential with offsetting effects of calcium entry and, for the most part, potassium egress. Then when the calcium entry ends around here, then we start seeing repolarization under the dominant effect of several different potassium channels as shown here. And then we get back to a polarized state, repolarized state. We begin again. One of the things you may remember is that we have absolute and relative refractory periods as the cell is repolarizing. Okay, so back to this again. Now I'm going to remind you that when we have those exciting impulses, they depolarize the cell membrane, as shown here, the sarcolemma. And then the next action that occurs is that you have a voltage-sensitive calcium channel that opens when there's that depolarization. So you have action potential wavefront reaching the cell membrane, this excitable tissue depolarizes, and that in turn activates the L-type calcium channel. I want to take a moment also getting past this oversimplification of what a T-tubule looks like, because this is what T-tubule looks like in a fragment of a cardiac myocyte. This is the T-tubule network. And I think this illustrates quite well how the function of these is to get the surface membrane into the interior of the cell to activate all those different myofibrils and to release calcium from all those different intracellular stores throughout the interior of these large cells. And so this is just the tip of one cardiomyocyte cut off here. But you see there's a lot of penetration of T-tubule invaginations of the cell surface membrane into the interior of the cell. And so, and this is just a fraction of a single cell. Okay, so back to the L-type calcium channel. It's a voltage-gated channel. That's the most important thing to recognize. So that depolarization event from the action potential and the sodium current influx triggers opening of this ion channel. It has regulatory subunits. I'm not going to go into those today. And it's, again, on the cell surface. In fact, its location is particularly dominant in the T-tubules. There are some L-type calcium channels on the sarcolemma that's not within T-tubules, but there's far, far more within the T-tubules because of their role. And their role is to link to the ryanodine receptor that's located in the sarcoplasmic reticulum. So a little more about the sarcoplasmic reticulum. It is an intracellular organelle. It's the endoplasmic reticulum of cardiomyocytes. It's the rough ER of cardiomyocytes, but in this cell type, its main job is to store calcium and large amounts of calcium. The amount of calcium that enters during depolarization of the cell and opening of the L-type calcium channels throughout the cell surface, the plasma lemma, is a small fraction of the amount of calcium that's sequestered into the sarcoplasmic reticulum. And as shown here on the left in skeletal muscle, there's a direct mechanical linkage of the L-type calcium channel and the ryanodine receptor, so that opening of this channel invariably causes activation and release of calcium from its intracellular stores. But here it's a different process in the cardiac muscle and in neurons. We see that you have a separate control of the voltage-sensitive calcium channel. It releases a little relatively small puff of calcium in this confined space that we call the dyadic space, the dyad being the L-type calcium channel and the ryanodine receptor. That small amount of calcium released in that localized area triggers opening of this calcium-sensitive channel, and it's a calcium-sensitive calcium release channel. And again, that's the ryanodine receptor. So as a result of this calcium activation, you get far, far greater, orders of magnitude greater calcium released from its intracellular stores inside the cell. And so this process then in total is called calcium-induced calcium release. And that is a feature that's specifically characteristic of cardiac muscle rather than skeletal muscle. I mentioned that the distribution of these different channels, right, is non-uniform on the sarcolemma. So this is the cell surface. This is within the T tubule. What we see is that there's lots and lots of L-type calcium channels within the T tubule, and they're directly opposed to the ryanodine receptor because that's their role, is to trigger that calcium-induced calcium release, and their spatial orientation is a good fit with that. Okay, so then we have the next phase of calcium-induced calcium release. We've now released a lot of calcium into the sarcoplasmic reticulum, right? Calcium has entered here, but most importantly, it's entered here in the dyad, releasing lots of calcium out of the ryanodine receptor into the cytosol, where it's available to activate myofilaments. And so now we've gone from the action potential part of this to the calcium transient part of this in red. And so now it's available to activate the myofilaments. I want to remind you that the myofilaments are mostly comprised of thin filaments and thick filaments. The thin filaments are thin, 5 to 7 nanometers in diameter, comprised mostly of actin, 2 strands coiled together, as shown down here, but also tropomyosin, a rod which tends to stiffen actin and also tends to either reveal or hide the actin binding sites for myosin. And then it's also comprised of tropomyosin complex, which is 3 peptides. One binds to actin, one binds to tropomyosin, one binds calcium, that same calcium that just entered the cell. And then the thick filaments are bigger in diameter, of course. They're primarily comprised of myosin, which has these long tails and these globular domains that eventually form the cross bridges. There's quite a bit more myosin molecules in each thick filament. They're arranged in a package as shown here. They have ATP binding capacity and also ATPases. And there's a new myosin head every 14.3 nanometers of each myosin head, but there's lots of myosin heads in a thick filament bundle as shown here. So let's take a moment now going over the process by which that calcium that was released via calcium-induced calcium release, much larger amounts of calcium in the micromolar order of magnitude into the cytoplasm. The first event is that there's calcium binding to troponin C, which is the subcomponent that binds calcium, and that induces displacement of tropomyosin. So in this resting stage, tropomyosin is hiding, for all intents and purposes, the actin binding sites that the cross bridges from myosin would like to bind to. Calcium binding to troponin induces displacement of tropomyosin and opens up those actin, the myosin binding sites or actin binding sites. Oops. So then we start getting engagement of the cross bridges with the actin binding sites on the thin filaments with ATP hydrolysis. From ATP getting converted to ADP, because this is a work stage of the about to begin. And that allows the power stroke that moves actin, let's say this way, apologize, relative to myosin. And this is happening in millions of myosin heads and actin binding sites throughout each individual cell. It moves actin relative to myosin, and that's the power stroke. And then actin is released. There's ATP binding again for the next hydrolysis event. Myosin, it gets cocked back into the pre-firing position. And then this cycling goes on and on and on continuously as long as there's high levels of calcium in the cytosol. And that once the cytosolic calcium decreases, Then tropomyosin goes back to its prior position of blocking the actin binding sites, and we can start having relaxation. So this is the final step in the calcium-induced calcium release, that active cross-bridge cycling, more than one cycle for every contraction of the heart muscle, many more, is continuing as long as the calcium's high, and is stopping and slowing and stopping once the calcium gets lower. So now we're back down to this little embedded diagram. We have the action potential coming first. That's the trigger electrically. Then we have the intermediary, the calcium transient, and that in turn, because of that troponin C binding and the cross-bridge cycling, begins to induce a contraction. And then as calcium is taken back up, we start getting the relaxation. So let's talk about, where does the calcium go after it's been released into the cytosol for long enough? Well, when the cell membrane repolarizes, then that'll close the L-type calcium channel and allow, and so there'll be no further influx of calcium at that point. In addition, there's an ATP dependent calcium pump called the sarcoplasmic reticulum calcium ATPase. It's a mouthful, but we call it SERCA, and the isoform for cardiac muscle is called SERCA2A. And SERCA2A is a powerful pump that takes calcium back into the sarcoplasmic reticulum. As you recall, most of the calcium that wound up in the cytosol came from the sarcoplasmic reticulum. And not surprisingly, this is a very high conductance pump that is able to take most of that calcium back into the sarcoplasmic reticulum in time for the next beat and the next release episode. There is a small amount of calcium, though, that doesn't go back into the SR, and that gets extruded from the cell via the sodium-calcium exchanger. And that extrusion is favored by the repolarization of the cell membrane at the end of the action potential. So in steady state, you can imagine that there's a certain amount of calcium entering the cell via the L-type calcium channel, and at the end of the cycle, that has to be extruded via the sodium-calcium exchanger. But there's a much larger amount of calcium that got released via the ryanodine receptor in the process of calcium-induced calcium release, and that got taken back up by the sarcoplasmic reticulum calcium ATPase, SERCA, back in there. And then the steady state is that those two processes balance, all right? Into the cell, balances out of the cell, out of the sarcoplasmic reticulum, it's balanced by back into the sarcoplasmic reticulum. So to summarize then, the stages of excitation-contraction coupling in cardiac myocytes are bulleted here. We have cellular depolarization. We have extracellular calcium entry via voltage-sensitive L-type calcium channels, LTCC, release of large stores of intracellular calcium from the sarcoplasmic reticulum via the calcium-sensitive ryanodine receptor, and that's the essence of the process of calcium-induced calcium release. So this L-type calcium was voltage-sensitive, but the ryanodine receptor is calcium-sensitive. The increased intracellular calcium binds to troponin C and activates myosin-actin cross-bridge cycling. And upon cellular repolarization, active calcium reuptake via an active transport process and the molecule sarcoplasmic reticulum calcium ATPase, SERCA, takes back most of the released calcium back into the intracellular stores, and calcium extrusion via the sodium-calcium exchanger is generally balancing the amount of calcium that came into the cell via the L-type calcium channel. To pull back and think about things that we think about as clinicians more often, and that is, we don't just have one contractility need for any muscle, whether it's skeletal muscle or cardiac muscle. We need to grade the degree of force to the circumstances. Now, in skeletal muscle, you may recall that they're electrically isolated from each other, skeletal muscle cells. And so there, the force is graded by the number of motor units that get activated, and there's a nerve for every muscle and a muscle for every nerve, and the more nerves that are activated, the more motor units that are activated, and the more force regenerate. In cardiac muscle, this is a functional syncytium. All the cells are electrically coupled to one another, at least within any given chamber. And so that means they are by design all activated together. We understand that from the use of cardiac resynchronization therapy, that when they're not all activated together, we get inefficiencies of energetics and contractility dynamics that impair pump function, and that when we restore that with something like CRT, we get improved pump function. So we know that we need to have a functional sensation within each chamber, but it means that if we're going to grade contractility, then we need to do it by something other than the number of motor units. This is electrically all coupled. So how do we do that? So one we're all familiar with, right, is the Frank-Starling curve, right? That we know that preload, which at the organ level is the amount of left ventricular end diastolic volume, and at the cellular level is the sarcomere length. We know that that is associated with an increase in intrinsic contractility that it grades force. So one way of that force is graded is by increasing cellular length, or at the organ level, stroke volume is dependent on venous return in the Frank-Starling mechanism. So that's one source of grading contractility. Another is something called the force-frequency relationship, which we don't think about very much, but if you study at the isolated myocyte level, you can, or isolated muscle strip level, you can demonstrate it. And the concept here is that as you have high stimulation frequencies, like you get when you're exercising, let's say, you get increased stimulation frequency, you get more calcium influx via the L-type calcium channel because it's opening more times per minute. And then remember, on the way out, the sarcoplasmic reticulum calcium ATPase and the sodium calcium exchanger are both either taking back up or extruding calcium, and there's a bit of a functional competition going on. And under normal circumstances, in larger mammalian hearts, the circa pump activity is quantitatively larger than the extrusion capacity for the sodium calcium exchanger. So bit by bit, the sarcoplasmic calcium load gets larger, and you have more calcium sequestered there to be released at the next heartbeat. And so over the course of, let's say, a minute of increased heart rates, you start getting increased calcium load within the SR because the SR keeps outcompeting the exchanger, and you get more released for the next beat, and you get higher contractility. So this is called a positive force-frequency relationship. Increased sarcoplasmic reticulum uptake kinetics, when they exceed exchanger extrusion kinetics, lead to increased contractility at higher heart rates. This is one of the things that gets abnormal in heart failure, but we won't go into that today. Finally, we're all familiar with beta-adrenergic stimulation, right? And beta-1 adrenergic stimulation in particular, that's what's the receptors that are most prominent in cardiac myocytes, and that they trigger a bunch of phosphorylation events. And the things that are getting phosphorylated are things that we've just been hearing about in EC coupling, which is why I wanted to include this here. The L-type calcium channel and its conductance of calcium is regulated by L-type calcium channel phosphorylation, so that when there is AMP-dependent phosphorylation, there's a bit greater conductance, and so you get a bit more calcium coming in for that calcium-induced calcium release. Likewise, in the sarcoplasmic reticulum, calcium ATPase is linked to another protein that I didn't mention called phospholamban, which has a restraining effect on the amount of calcium reuptake into the sarcoplasmic reticulum. But when it gets phosphorylated, it has less restraining effect, and you get more calcium uptake into the sarcoplasmic reticulum as phospholamban gets phosphorylated. The ryanodine receptor 2 is phosphorylatable, and it has even higher release, so that you have more calcium coming in, you have more released for the calcium-induced calcium release, and then you have more stores of calcium within the sarcoplasmic reticulum, and all of this tends to favor bigger calcium transients. The one thing that doesn't go quite the way we'd expect is that when troponin is phosphorylated, you actually get a reduction in binding of calcium. And that doesn't matter so much, because at the same time this is happening, there's so much more calcium entering the cell via these blue events. In fact, this facilitates relaxation, that basically the reduced binding of calcium when troponin I gets phosphorylated leads to a faster off response that allows faster relaxation, which of course we need when we're going at faster heart rates. So the whole summative effect of this adrenergic stimulation is a number of phosphorylation events of key intracellular calcium regulatory or calcium responsive proteins, and all of them favor increased cardiac output. So just to review, again, the macro purposes of all this, right? We know our heart must pump continuously throughout life, and we have intrinsic pacemakers, which were not a major focus of this talk. We know that the whole muscle must contract with each beat, and we have a specialized conduction system and gap junctions to make this a electrically functional syncytium. All cells are electrically linked. We know the heart muscle must relax with each beat, and that's one of the reasons we have a prolonged action potential with that long plateau phase and electrical refractoriness, because we need time to refill. And then the heart must adapt quickly to changes, and that's what I was just highlighting. If we don't have the ability to grade contractility with individual motor units in a functional syncytium, then we need to grade contractility with preload, heart rate, or beta adrenergic stimulation stress. And of course, these are linked, right? The same beta adrenergic stimulation that causes all those myocyte phosphorylation events also increases the heart rate at the same time. So I'll stop there and be happy to take questions.
Thank you, Dr. Margulies. That obviously such a complicated topic, and I think for me personally, I think that was the best that I've ever understood it, with having you talk us through those steps, because I know when I've tried to read that through textbooks in the past, it's been very difficult to understand and very complicated. So I appreciate you doing this. Are there any questions from the listening audience? I know we had a few people jump off already at the top of the hour, but if anybody has any questions, feel free to mute yourself, or you can drop it in the chat.
I do want to mention these videos. They're nice if people want to see more presentations rather than hearing mine again. These are really good, and they go into other topics within and beyond this.
All right. Well, thank you so much. And I guess then we'll just go ahead and wrap up for today. So again, this video will be available on our YouTube site for those that want to go back and watch it again for a second time, and to take a deeper dive into all the mechanisms that Dr. Margulies so eloquently described for us. So thanks again, and I hope everybody has a great rest of their Monday.
Okay. Thank you.
Bye-bye.
Good luck, everyone. Bye-bye.